You hear me over there? Is the sound good? Okay, perfect. So indeed, my goal is to tell you a little bit of how do we develop a network perspective for aging. And why do we need that? Because much of the aging research and much of biology research is really based on genomics. But genomics is often, I say, it's a little bit like getting the names and the addresses in the phone book of a city. But you need a Google map to really see how the pieces are connected to find anyone. And in case of the cell, the Google map is something like this. So what you actually see is this one is a protein-protein interaction network, which is kind of the sum of all the physical interactions that were experimentally documented within the cell. And so two proteins here are shown as having a link if they bind to each other. And the reason why this is actually interesting for us is that, and of course, beyond the protein interactions, you can also consider protein DNA interactions and many other regulatory interactions and so on. This particular visualization has only the PPI network. But the question is, how do you conceptualize the disease within a network like that? And that's really what my lab has been focusing on in the last kind of 15 years, and now became kind of a subfield called network medicine at the intersection of network science and medicine and statistical physics. And so one of the key concepts, at least for me and for many of us, is actually the concept of the disease module, which is the finding that when you take a particular disease and you kind of identify using genetic tools, what are the genes involved in that particular disease, and you place them on the network, what you realize is that the nodes or the proteins are not randomly distributed within that giant network space, but they tend to be aggregating in a certain network neighborhood. And this of course makes sense because a disease typically is the breakdown of a function. A function involves many interacting components, and the interactions mean that they are kind of close to each other in the network sense. And then network medicine has kind of developed a set of tools to identify these disease modules and also to go a step further and kind of identify who are the other proteins who may be involved in the disease because they are in the proximity of genetically identified disease genes. And these are very important. It's important to go beyond the disease genes because many of the drug targets are not the disease genes themselves. We cannot fix the problem except if we use genetic engineering. But we tend to instead kind of act downstream, so the target has to be downstream from where the problem is. Now, network medicine has offered a set of tools, and the most relevant, I think, for us, at least for my talk today, will be the concept of drug repurposing or drug target identification. The idea is that could we use the network to identify whom do you want to target in case you want to target a particular disease or cure it, right? And the concept is shown over here. So this is a schematic picture of what I showed earlier. Nodes are proteins or any cellular components, and the links correspond to physical interactions. And it's important we always focus on physical interactions because that kind of hides a mechanistic explanation if you find an answer there. And here I'm highlighting two neighborhoods. These are what I would call the disease modules for two different diseases who in this picture happen to be nearby, but they're also distinct. And the idea that if you want to find an effective target, then what you're looking for is targets that are either typically within the disease module, and we've shown it actually many times that the drug targets are within the disease module. And if you think about drug repurposing, that is finding drugs that are already on the market that could be repurposed for that disease, you are again looking for drugs that have a valid target in that particular neighborhood. It's not as simple as bean counting that I need to have a target in that neighborhood. We have statistical measures to show whether you are hitting in the right neighborhood or not. Now, what we want, this kind of picture has been very useful, and many people are using it to identify drug targets and drug repurposing opportunities for many diseases. And what in the last 2 years we've been focusing on is that could this be applied to aging? And the starting point, of course, always of this story is to find genes that are involved in aging. And thanks to the community around here, we know that, right? So there is the concept of hallmarks of aging that I don't need to introduce in this particular audience. But the most important thing is that as of last year, we have actually a whole set of genes that have been associated with each of the hallmarks. So on the left-hand side, you see the traditional hallmark picture. On the right-hand side, for each of the color over there, we are actually showing how many genes had been associated with that. And the stars correspond to the degree of the evidence, which is not assigned by us, but assigned by this particular paper on the open genes that classified genes based on these things. Now, the first question you would ask is that how does a disease module look like in the case of aging? And we are treating each hallmark as a separate disease, if you want it like that, or a separate phenotype. And we're asking, do we have a disease module for each of them? And the nice thing here, actually, this is kind of more a statistical picture, and I'm just going to tell you what it means. For each of the hallmarks, there is a very well-defined statistically significant disease module, meaning that the hallmark genes tend to be aggregating each of them in their own network neighborhood. So that is as good as it gets when it comes to network medicine. We don't always get it. We get it lately for many diseases, but it's not guaranteed that this works. But for all of the hallmarks, actually, we do have a very, very distinct disease module. And the question is, how do these disease modules relate to each other? How do the hallmarks relate to each other? And for that, we have a concept developed many years ago of where are two diseases in terms of the network. Here I'm showing you actually asthma in red and COPD in yellow. And these have very similar symptoms, even though they are distinct diseases, and yet they tend. So not surprisingly, they are in the same network neighborhood. And we do have a measure that we developed a few years ago, almost 10 years ago, which is kind of the disease distance in the network sense. So we're trying to figure out how far are two disease modules from each other. And this is a very elegant, simple statistical measure that measures the distance between the genes involved in the two diseases. And this measure is negative if the two disease modules are overlapping, and it's positive if they are not, that if they are the different network neighborhood. And that is important because we have shown in this science paper that if two diseases are overlapping, then they have high comorbidity, high co-expression between their genes, and so on. And those whose distance is 0 or any positive value, they have nothing to do with each other in the network sense. So it's an important measure to see how far they are from each other. So how do these kind of diseases relate to each other? How do these different longevity modules relate to each other, or the hallmarks? Well, some of them are more isolated than the other one. We have actually here a group of them that are very, very overlapping, and we have some that are actually like epigenetic variations and changes in the extracellular matrix structure that are somewhat isolated from each other. So you see some hallmarks in the network sense are really in the same network neighborhood affecting similar processes, and others kind of stand on their own in a separate world. Now, where do we go with next? Well, we would like to now find out, we said, let's identify drug repurposing opportunities for each of the hallmarks, or could we find some that actually could affect multiple hallmarks at the same time? And for that, we ended up pulling in a pipeline that we were somewhat lucky with the COVID that we could actually validate at a very, very large scale, meaning that COVID offered us for the first opportunity to take a disease, in this case an infectious disease, and effectively test all the drugs out there and see whether our tools are working or not. So during the COVID, we ended up doing this big project that we made in a month, between March 23rd and April 15th, we applied all the drug repurposing pipelines, the network-based one, for COVID. Then we froze the pipeline, and then we started working with the experimental colleagues to test how well our repurposing predictions work. At the end, we only tested a few hundred at first, but then at the end, we ended up testing 6,000 approved drugs. And we now kind of have a good confidence of which pipelines and how to use them. We're bringing that to aging, and we're asking the question, are there drugs that could be hallmark specifics? And those are in the top, right? So like this particular drug is really affecting only that hallmark, and that one is that. But we're also looking for multi-hallmark drugs that have that hit in the neighborhood of multiple hallmarks, so in principle could perturb multiple network neighborhoods and could affect many processes simultaneously. And of course, we did end up applying actually this particular pipeline to the whole aging field, to the aging modules. And this is just to kind of show you how the results are kind of look like and where we are with the process. So here's I'm showing you, for example, one drug. I'm going to show you the top 5 in this case. This one is actually has rank 35, means that it actually affects 35 sub-hallmarks, meaning that at different levels of confidence, the colors, the hallmark significance tell you which hallmarks are hit by that. This drug has not actually been tested ever for aging, right? So there's no animal models, and I'll tell you about CMAP significance and so on. And in principle, we do get a rank list for virtually all the drugs, right? And the question, how do we know that we are right? How we do know that at least these predictions make sense or not, because they're effectively based on the network? Well, on one end, of course, the previous experience with other diseases, but then we can actually use expression data to say, do these drugs that we are predicting to be effective really affecting the disease module or the hallmark module? And here I'm showing you 2 examples to see how this works. So the first on the left-hand side, anisodamine, which is actually a drug approved in China for circulatory shock as well as myopia. We predict to be far away from aging, right? So it should have no effect. And when you look at the expression changes of the cell lines that were given this particular drug, you actually don't see any major modifications of that. So like this drug is really not affecting this particular aging module. However, when you look at cerulatin, which is another drug, which is an antifungal antibiotic, we predict that has proximity 1, means it is very, very close to the disease module, hitting there. And indeed, we find that most of the genes are kind of over or underexpressed subsequent the drug to be given to the cell line. So this is one way that you can actually test these predictions. And then we now have this hit list, and we're hoping together with Vadim Gladisev lab to go and do more in-depth validation, both from cell lines effectively and then going to animal models whenever it's possible. But what I would like to also mention that it's not only drugs, right? But we're also always looking for new molecules that could affect, that may not be already on the market as a drug. The advantage, of course, drug repurposing is that you know already the toxicity, you know what the drug is doing to you, so it's a much faster, if it works, it's a much faster process to get it to patients, right? But we're searching for other low-hanging fruits, and that is related to a journey that we've been having in my lab for many years now, which is the foodome project, which is the idea that could we actually do interventions, in this case, into aging via food molecules. And why food? Well, because we know that diet does modulate longevity. If nothing else, everybody knows that you should all be eating Mediterranean diet, right? And you should be all kind of avoiding the Western diet because they clearly have very different impact on longevity. So the question is this just a whole diet, or are there specific molecules within the diet that affect effectively aging, and could we identify what are those molecules? And of course you would say, well, then it's a nutrition problem. And my answer is no, has nothing to do this with nutrition. And the reason, and I was myself very surprised when we started the foodome project of how little nutrition addresses in this story. Because we thought when we started this, well, let's just take the nutritional components and see how they influence the network. And then we were very surprised to realize that nutrition is really focusing on 150 molecules within the food. This is what is often called nutritional components or nutrients. They tend to be sources of sugar or sources of energy. And, of course, they tend to be the vitamins. These are the stuff that you must have to survive. These are the essential components that you need for maintaining maintaining life. But then we realize that there are many, many other components of the food or chemicals in the food that are not nutritional components, and we ended up calling it the dark matter of nutrition because no one tracks them. And as a result of a 5-year project, now we have 135,000 molecules within my lab that have been identified with one or multiple foods. And so, for example, if you take garlic, if you go to the nutritional database, it will tell you that it has 67 nutrients, but we have 5,600 other chemicals that are in the garlic, which means that every time you eat something, more than 10,000 molecules through that dish are bombarding your cellular system. How do they do that? Well, the reason why they were ignored because we assume that they don't have a metabolic role. The metabolism doesn't engage with them, hence they are irrelevant. They go in and go out. Now we know better because one of the biggest class of these chemicals are the polyphenols. And just to give you an idea of how they look like, so if you take sweet basil, right, we have about 2,100 chemical compounds, of which about 500 are actually, well, 800 are flavor components, they're volatile. And then about 300, and many of them are actually already have a disease association that we know that they're involved in disease. How are they involved in disease? Well, polyphenols is a good example. We know that we're supposed to eat lots of fruits and vegetables because they have polyphenols that are good for us. What do? They're not processed by our metabolism. They're a little bit broken down by the bacterial metabolism, but they get into the bloodstream and they bind to proteins and DNA and they modulate the activity of the cell. So they have a regulatory role and not a metabolic role. And so we have these 135,000 molecules approximately that regulate the processes within our cells. And some of them are good for you, some of them are bad for you. They come in small concentrations, most of them. So we have to identify which is good and what is bad for you. So how do we do that? Is the same process as drug repurposing. But now we're not looking at the drug and its targets, but rather looking at the food molecule and where can it bind, to what human proteins does it bind. And we realize that, yes, there are about a few thousand targets of molecules that have a known target, that we know what proteins they bind from experimental measures, but we don't know for all of them, so we end up developing this AI bind tool that effectively identifies where these food molecules would bind using the smile of the molecule and what human proteins they can bind to, and even tell you for roughly the pocket where they are. And then the question is, once you know where they bind, then could you actually identify what is their potential mechanistic or therapeutic effect? And we have done that a few years ago in Nature Food for polyphenols, and we took it all the way to the end that identified polyphenols that we predicted to have an effect on platelet formation. And then our colleagues at Harvard ended up doing the experiments, and they confirmed that indeed the effect was there. So now we are actually bringing this tool set to aging, and we're trying to identify which of these 135,000 molecules could have an effect on any of the hallmarks, not just like the drugs do, or multiple hallmarks. And of course, the advantage that if we find any of them to have a true effect, then, and there will be probably many, you don't necessarily need FDA approval for that to begin with, because they are naturally occurring molecules within the food. So you can identify either diets that are kind of good for aging, but also you can also go ahead and modify these molecules if you want and turn that into drug. Now, what I would like to finish with is a couple of other projects that we're pursuing in the lab in the spirit of these problems and network medicine. And one of them is actually with Jan here. We actually have a wonderful collaboration of looking where the mutations occur within actually the cell and in the network medicine perspective. And we have a very surprising result that we're just writing up now, because if you look at how many mutations each gene has, and these are semantic mutations that you acquire during your lifetime, it turns out that you have roughly the same number of highly mutated or lowly mutated genes, right? So it seems to be random. But when you actually look where they are in the network, it turns out that the lowly mutated genes are all in specific pathways, and you don't find highly mutated genes in any of the biological pathways. And the same for disease modules. We have only 3 disease modules out of several hundred that contain highly mutated genes, and most of them are actually enriched with lowly mutated genes. So there seems to be perhaps an evolutionary driven, I don't know, like some kind of enrichment that is really kind of protecting the genes involved in the pathways and in the disease modules from mutations, and it's just ignoring the rest of them. So the highly mutated genes seem to be randomly distributed in the network. The lowly mutated genes are all in the well-characterized pathways and disease modules. If you want to learn more about it, Joey here somewhere, there it is in the back, he will have a poster on Wednesday talking about some of these results. And in the spirit of Uri's work, we also have equations. We're developing a network dynamics of aging, which is based on the work that we have done a few years ago, where we developed a network perspective towards resilience. The resilience idea that the system can break without actually being broken into species. It's kind of not able to recover from perturbations. And turns out that whole tool set is really amenable to aging problems. And we're trying to actually figure out what is the relative role of the mutation accumulation as well as the epigenetic damage. Because when it comes to this kind of continuum equation that describes the resilience of the problem, they seem to be affecting different terms, so they can be really nicely distinguished from each other. This is very much work in progress, but we're very excited about that. And finally, I should just kind of show our collaborators, the team, of which Joey is here. So if you want to talk to him, he can actually tell you more details about our work. And we have our wonderful collaborators, from which Vadim and Jan are actually also at this conference. And I'll just finish with this network, because it's all in the network, right? If you want to take one message away from this talk, if you want to understand aging, the journey will go through this particular network. Thank you very much for your attention.
Beautiful, thank you. I think it's also the network that we're building here that's very important. So I see some hands, we have times, questions. One over there, one here, then we move down the room.
Hello, thanks for a good talk. In the context of these hallmarks or disease modules in your network, I'm wondering about how you define them. If you only do it in a network native or a graph native way, or also in an embedding space, and then if so, what kind of embedding methods do you think hold the most promise?
So we've been doing this way before embedding became very fashionable. So all the tools that we've been using traditionally had to do with just the network space. So the measure that I showed you is all purely network space. And the reason we do that is because if you have a prediction that this drug is hitting here, that's a mechanistic prediction that can be immediately moved to the lab and can be tested, because we know where the changes in the network should occur if we are right. Now we live in the 21st century, in the age of AI. Now we have also lots of tools available to us that are AI-based. So far, the predictive power is comparable, and we don't always know which one works better, whether the pure network or the AI one, so we kind of throw all the battery on that. The problem with the AI-based tools is that it gives you a correlation and not a causation. Not that the network gives you a causation, but the network gives you an experimentally testable hypothesis. In the case of the embeddings, we don't get that, but we use them to as a comparison always.
Other hands are up, one there. Oh, sorry, yeah, you
Okay.
yeah,
Go for it.
Thank you for this very insightful talk. And I would like to know more about the model, and especially in terms of dose. So no matter for drug or for food,
yes.
So we can see that dose is very important, and we can assume that's a U-shape association. So how do we evaluate from your network?
Well, that's a very good question. For drugs, we already know the dosage, which is toxic and so on. So, and for food, it's a very interesting question, what should be the dosage? Because when you look at the concentration of the food molecules in the food, they span 9 orders of magnitude. Meaning that water is the most prominent in food, which is kind of 70% of 100-gram food is water, right? And then vitamin A is 10 to the minus 6 grams per 100 gram, right? So there's a 9 order of magnitude. So on one end, you can ask yourself, what is the natural concentration in which we actually achieve that? And we know that, right? Because we know what's the concentration of that chemical in the food, and we know that's safe for us because we're eating it, right? But what would be then what could you actually do as a drug if you increase the concentration? That would be a one-by-one conversation when you try to put this on the market, right? Because obviously anything is toxic in high enough concentration, including water, right? So we know the natural range, and we know also that the natural range from one food to the other one, it changes about 1 in an order of magnitude, so not more. So like vitamin K will never become 1 gram per 100 gram, right? It goes from 10 to the minus 6 to maybe 10 to the minus 5 in the most vitamin K dense foods, right? So the variation is relatively narrow, 1 in order of magnitude. So we could use this as a guide that's safe range, and if you want to go beyond that, that becomes a toxicity and a safety study. Mm-hmm.
Yogo.
Hi, great talk, thank you. Actually, about 5 years ago, I tried to use your method of the distance metric in the networks. And as a biologist with RNA-seq data, I actually faced a limitation, which was eating a module is not sufficient. You actually have to eat it in the right way because of the fold changes going, up or down. And at least back then, the method was more, does it eat or not this node?
What do you mean eating the node? Hitting or eating?
Hitting, sorry, that's my English. So it was basically a 0 or 1, but in fold changes, the direction of the fold changes are very important because we don't want to aggravate the hallmarks of aging.
So that's a very interesting question, right? Because like the way I'm hearing you is that you're thinking about a single target and what is the impact of that, right? We have just finished a study that we never published, but I mean, we may publish it, where we actually tried to say if you just consider the therapeutic target of the drug or all the targets, which one works better for predicting existing drugs that would work? And you have to consider all the targets. So when you just consider the therapeutic target alone, for one reason that we're still trying to struggle to understand, the prediction is not as accurate as considering all of that. So if you're looking at a single target, single drug, right? One hypothesis I have is that many of the drugs that are on the market, which we're using as benchmarking, do actually activate multiple things, even though we have one therapeutic target that we kind of believe on and swear on, right? So maybe that's a question, is that did you apply to a single one or to multiple targets?
No, actually to several, because for example, rapamycin in C. elegans changed about half of the transcriptome.
Okay.
The question would be, actually the fold changes matter. So there are genes that go up and down with age, and just eating that module is not enough. Because we might be eating it the wrong way.
Okay, and in that sense, yes, that's a whole different story, right? But we're dealing with that in the case of the CMAP story, right? And not all drugs that are known actually or are tested currently for aging do give you a positive hit. I have somewhere a table for that. Okay.
Really amazing. 4 more hands up. We go Raghav, then the guy in white, then the lady behind on the left, on the gentleman here in red.
Yeah. Really amazing.
Get the algorithm?
Okay. Raghav first.
Yeah, Albert, really amazing presentation. I might steal your PPI network. Please make it public.
It's not a stealing. You know how they say that? What did Picasso say?
It's a beautiful visual, I would say that. One question that I have for you is, you had a very nice explanation around sort of specifically your aging modules and the representation of the hallmarks of aging. The question I have is the underlying data that's going in. Is this in vitro, different cell line data, or is this in vivo data from animal models or human in vivo data? What kind of data it is?
So the binding information is really chemistry, right? And that is universal across organisms and across species. So that's the starting point. So what you see actually is the skeleton of the network.
Right.
Because all human proteins in this particular image and all the binding. Now, if I'm interested in brain, I throw out all the proteins from the network that are not expressed in the brain, right? So you do need to kind of make it cell-specific. And now with single-cell measurements, our job has become much easier to kind of restrict them. And then if you do that, and that's the beauty of it, right? The question is, why don't we get osma in the brain? right? And because many of the genes involved in asthma are actually expressed in the brain, but not the whole module. So a few of them are not, and hence the module becomes disconnected and you don't have an asthma module in your brain, you only have in your lung. And when you look at the brain tissue, the only disease modules that are statistically significant are related to brain disease. So just a long story to say, short to say, we have a way of restricting that, but this is kind of the overall architecture, and when you care about a particular cell line, you need to go and restrict it. And if you have localization information, you could do that as well, because if two proteins don't meet, even if they could interact, whether they met, if they don't meet because there are different compartments, you can eliminate that link. But that comes at the later stage of the story, right? Once you kind of started to narrow down a particular disease, particular tissue, and so on, then you can start doing this fine-tuning in the problem.
Specifically for the hallmarks data, what did you use? I was curious, specifically for the hallmarks data.
We haven't done that for the hallmarks data. What you see is based on the full skeleton.
Got it. Okay, thank you.
Sure.
Gentleman in white.
So aging is a loss of, over here.
Okay, perfect.
So aging is a loss of biological function,
Uh-huh.
so a loss of, you could say, in your model, a loss of network function.
Yes.
Then a problem with using the hallmarks is that you use, I know, like I understand why you use them, is that you use the functional output of this consequence.
Yeah.
When you talked about network resilience, is it also possible to use your model in a neighborhood distance to see how resilience is lost with age?
Yes.
And then see where your drugs could be targeting in this neighborhood?
Absolutely
And are you
right.
doing this?
Well, we will do that probably, but what I want to kind of emphasize is not only aging, but all disease is loss of something, right? We don't know, not even with drugs, how to create new links, right? All drugs are blocking existing links. So at the end, I think you are saying something very profound, but it's more general than aging, right, per se. And then you're right, when we're going to start thinking, when we get to actually put it on the real network, the resilience framework, we don't necessarily need to limit ourselves to the hallmarks. We could, in principle, apply to the full network and see if it gives us any information that is useful. We're not there yet. We're more on the conceptual side right now.
Person
Hi, Professor.
white, yep.
Yeah. Wonderful talk. So I'm from Oxford, from University of Oxford. So my work focus on aging and leukemia. So I'm just wonder if there is some network medicine of disease. So if you compare the network medicine of disease with the network medicine of aging, if there's some part they are very close to with each other, whether this can indicate the and aging. So if you rejuvenate aging, whether can avoid this disease and also for the disease treatment.
So I missed the key word that you said. There is a network medicine of the
disease, some disease, for example, cancer, like this. Yeah, because, during aging, also some disease came out.
Yes.
So if we know the network medicine of the disease at the same time,
I see.
and also you are very good at the network medicine of aging. So if we compare them together, find the close part, So
in principle, and that's a very good idea, I never thought of it, but we could do that. In principle, now that we have the disease modules for the hallmarks, we could see what other disease modules are overlapping and what they are not. And in principle, that should give us, if we are right, all the aging-related diseases. So that, in principle, should be possible. Okay?
Person in red, and then last person in the black shirt there.
Yeah.
Thanks for a fantastic talk. So you mentioned the food contained thousands of molecules spanning orders of magnitude.
Hundreds of thousands.
Hundreds of thousands. It was better. The safe presumption will be that these molecules are not act entirely isolated. I mean, vitamin C is a great example, right? It's ascorbic acid with flavonoids. So the chances that if you take one molecule out of that mix, they may not work.
Oh yeah, yeah. Oh,
and so how are you dealing? So how are you dealing with the combinatorial nature of hundred thousands of molecules per food, and
Are
each
you
thousand
trying
of
to
them
be
probably
really nasty with us now? I mean, this is why the beta-carotene studies have all failed, right? Because people find, if you eat beta-carotene-rich food, you're not going to have a heart disease.
And many other studies.
And then when they did that, they turned out no effect. And turns out beta-carotene always comes with 400 other molecules, right? So you don't know whether it was beta-carotene. Well, the beta-carotene is nice and orange and bright and so on, so it's actually a good target, but it's not doing what we thought it's doing. So first of all, you're right. They always come in batches, right? So it never comes a molecule alone. But we have mechanisms, right? So in a sense that we are hoping that we can avoid that trap by having the mechanism of action of each of the molecule that we kind of put forward. And so even if there are 300 molecules who come simultaneously with, if they don't hit in the same network neighborhood, for us it's not a signal. Do I make sense? So we're just using these 130,000 molecules as one drug at a time and not one food at a time. Do I make sense? So they are just inspirations for finding kind of drug-like things for us. Now, if we would actually have to go back and say, I'm going to make now eating recommendations based on that, then I would have to worry about what you say.
It's just the way they're combined could be reflected by the way this plant was grown. If you just put those 1,000 molecules next to each other in the water, nothing will happen, right? So that's
always a possibility, and that's what clinical trials are good for us.
Last question here.
Hi, it's actually a very basic question. What data do you recommend to use to build these PPI networks? Like, are we talking string database or something else?
There are many different ways. Our data is always available on our website. When we publish a paper, we put it out there. Some people use string. It really depends on your particular flavor. We update our data set kind of every summer. We have a kind of a week-long process to see what new data sets came along and what should be added and validation and so on. But some of my colleagues are just using strings and other data sets. It's always a personal taste. We only use experimentally validated interactions. We don't use predicted interactions per se. And we also have a way of weighting it of how much evidence is for that, of how many different times that has been seen. And that gives us a way of kind of saying that if you remove the weaker ones, are the results still surviving? But other than that, it really becomes a matter of taste. But if you want ours, get it from our website. Okay?
Perfect. Thank you very much.